First, how complicated is the brain? How complex is it as a machine? This is a question an engineer might ask. From the outside, perhaps, it doesn't look that complex. But as soon as we take a look inside, and here is, for example, a slice through the brain, we're struck by its enormous complexity. So this is an image from yet another kind of magnetic resonance imaging technology called diffusion tensor imaging, which shows the long-range connections between different brain regions. We're looking at just a single slice, and we're looking at connections, each of which represents thousands of individual axons. And I think you can appreciate that the wiring is very complicated. In fact, if we look at just some basic numbers regarding the complexity of the brain, we can't help but be impressed, as an engineer might perhaps, in looking at how sophisticated this machine is. So let's just ask, for example, how many neurons there are in a brain. Well, in a human brain, there are about 100 billion neurons. The average neuron in the cerebral cortex, for example, has about 10,000 connections to other neurons. So that is 200 trillion connections total in the cerebral cortex. If we ask, for example, what the total length is of all the axons, the long-distance wires that connect one cell to another, one brain region to another, the answer is, for a single human brain, it's about 1.5 times 10 to the 5 kilometers. That is about 5 times the circumference of the Earth. That's a lot of wires to pack into one brain. But that, in fact, doesn't tell the whole story. These numbers, impressive as they are, if we look at even a single neuron, we see that it is enormously complex. So here is one neuron that has been made fluorescent with a special dye that's been injected into it. And we see this enormously complex branching pattern with hundreds and hundreds of branches. This sort of pattern is stereotypical for a given kind of neuron. Other neurons will have different branching patterns. And furthermore, if we look at the packing of neurons, it's extraordinarily precise. So there's very little wasted space. Here's a picture from a mouse brain, a genetically engineered mouse, I should say, in which different neurons have been labeled with different colors. You can see they're very tightly packed. Their branches, which are coming out to the right side, are also very tightly packed. They're packed in with other branches that are not fluorescently labeled here. There's virtually no wasted space. If we were to compare the brain to devices that we humans have made, and this might be the sort of comparison an engineer would like to make, we can ask, how does the complexity of the brain compare to the complexity of our man-made objects? So let's take the most complex thing that we humans have made, a computer chip. This is a typical computer chip. There are perhaps 1 billion different elements that have been manufactured on its surface. It's very tiny. It's smaller than a coin. And if we look at high power, this is now an electron micrograph of the surface of the chip, we see that the elements, the individual elements on the chip, are about 50 billionths of a meter in diameter. So that's about the size of a synapse. So we humans have done pretty well. We've got the miniaturization of our devices down to roughly the size scale of the brain's devices. But I want to remind you that computer chips can only be manufactured in 2 dimensions. They're on flat surfaces, whereas the brain packs all of the machinery into a 3-dimensional space, and very efficiently at that. So the brain can pack a lot more circuitry into a little volume than a computer can, by many, many orders of magnitude. An engineer might also ask a question about how the brain is built, compared, for example, to the building of our man-made devices. Now, when we build something, we typically design it and draw out a blueprint so that we can have an image of the thing we want to build. Here's a piece of metal that's been machined. We can see that this is certainly a logical way to encode the information for the object that we're going to build, encode it essentially as a picture with commentary written on it. And if we have something that's very complex, like a brain, we might imagine that having blueprints, wiring diagrams essentially, would be very helpful. But in fact, nature does not use images to encode the information for building anything. The information is encoded, it turns out, in a long one-dimensional string of letters. It's along the DNA, our genetic material. So this is much more like text, where here, in this case, the alphabet of DNA has 4 letters, 4 chemical groups, which we abbreviate A, C, G, and T, and it's the order of those groups along the DNA chain which is the information. How information that is encoded in this one-dimensional form along a strand of DNA is used to produce objects as complex as synapses or whole neurons or whole brains is very much an area of active research. There are a number of advances that have been made, but I think it's fair to say that most of the story still lies ahead of us. And I therefore want to close with 3 open questions, questions that we neuroscientists are intrigued by, questions that are still largely unanswered, but in principle are answerable. And let's look at those one at a time. Question 1, how is brain structure and plasticity encoded in the DNA? It is encoded in the DNA in the sense that each organism, whether it's a person or a sea slug, has a particular kind of nervous system, and that nervous system is largely a product of their genetic inheritance. It is also a product of their environment in the sense that changes to the nervous system change the nervous system itself, and that, of course, brings us to the second question, which is what those changes are. What are the cellular and molecular representations of memories and thoughts? What happens inside your head when you hear a new phone number or a new name or see a new face? These are still largely unknown. And finally, how do genetics and experience interact to make each brain structurally and functionally unique? And this really gets at the heart of our own sense of ourselves as unique individuals. And I think the answer to each of these 3 questions, and especially this last question, is going to shape our view of ourselves, both as individuals and as a species. Thank you.